Hey everyone, my name is Brian. I'm one of the Bio107 TAs, and today in this video we'll be talking about diffusion and osmosis and how it relates to biological activity. Diffusion is the movement of molecules from an area of high concentration to an area of low concentration until an equilibrium is reached, meaning that the concentration of those molecules is the same throughout this entire space those molecules are occupying. And in your lab simulations, you'll see the word or the term concentration gradient, and that's simply referring to that difference in concentration. So if you have an area of high concentration and an area of low concentration, you have a concentration gradient that molecules will diffuse down. And we can see this in action in this picture here. So we have a beaker of water, which will act as our solvent. We have this diparticle mixture, which is our solute. And when we add the diparticles to the water, we get a solution. And we can see that initially these diparticles start out all bunched up in the same area in a high concentration. And so we have a concentration gradient where one side there's a high concentration and another side there's a low concentration. And so eventually those diparticles will want to disperse throughout that water solution until they're in equilibrium and equally distributed throughout that solution. And this is driven by something known as Brownian motion, which is the random movement of molecules. Basically, all these molecules are just bouncing off of each other and the sides of the container that they're in until eventually they're all equally dispersed within a given space, as you can see in this animation here. And this requires no input of energy. It's known as a passive process, and it can happen across semi-permeable membranes, which we'll talk about on the next slide. So a semi-permeable membrane is a layer that allows only certain molecules to pass through based on several characteristics, which I'll talk about in just a second. So here in this picture, we can see that there are two types of transport. There's passive, which includes diffusion, and facilitated diffusion, which is a little bit different. And then there's active transport. Active transport actually requires energy, whereas passive transport happens naturally without inputting any energy. And so in this example, this cell membrane is our semi-permeable membrane, only allowing certain molecules to flow in or out. And we have a concentration gradient where we have a higher concentration of molecules on the outside of the cell in the blue area than we do inside the cell in the tan area. And we're going to focus mostly on diffusion here. I just wanted to show you that there are different types of diffusion and the difference between passive and active transport. So whether or not molecules can diffuse across a semi-permeable membrane depend on molecule size. So in this example, the purple molecules, we could say that those are the smallest ones that are actually able to pass across this membrane, whereas the orange triangle and the blue circles are perhaps too large and they require these protein structures in the membrane to actually get them to the other side. The charge of the molecule matters. This has to do with the fact that the membrane is composed of these phospholipids, and they have an inner layer that is nonpolar, meaning there's no charge. And so any molecules that have a charge are going to be repelled and not able to cross through there. You can think of it like trying to hold together two magnets that have the same charge. They're actually going to be repelled. It's that same concept. And then finally, the membrane pore size matters. So if the spacing in between those little phospholipids in the membrane were larger, then perhaps the triangles and the blue circles would be able to pass through without needing these facilitating protein structures. So now that we've determined whether or not diffusion can actually occur, We can then look at some of the factors that will affect the rate at which it happens, or the speed over time. So one thing that can affect the rate of diffusion is the difference in concentration gradient. So if you have a really big difference where on one side you have a really high concentration, on the other side you have a very small concentration, you'll have a fairly fast rate of diffusion, as opposed to a solution where it's mostly mixed together, but maybe you have a little more of a molecule on one side than the other side, you'll have a much slower rate of diffusion in that scenario. Temperature also plays a role. So the more energy that you put in, the more you raise the temperature, you're going to have a faster rate of diffusion. And you can think of, boiling a pot of water. As you heat that up, those water molecules are going to start moving around faster. You're going to get more of that random motion, and the solution is going to get mixed up a lot faster. The size of the molecule is important. So in this scenario, we have cut out 2 holes in this agar gel plate. You can think of it almost like a Jell-O material, but it's much more firm than that. And then we've dropped in a small red dye particle into one of the holes and a larger blue dye particle into the other hole. And we can see that the smaller red dye particles have diffused much further than the larger blue particles, simply because they're smaller, they're able to get through those pores a lot more easily, whereas the blue, larger particles, they have a harder time moving through that material, and so it makes for a slower rate of diffusion. And also the density of the media that the molecule is in. So if we took those same dyes and put them into water, they would diffuse much faster than they would through the gel, simply because all the molecules in the water are much more spaced out. They're able to move around much more. So the density of the media will also play a role in the rate of diffusion. And so now that we have hopefully a pretty good idea of what diffusion is, we can talk about osmosis, which is essentially the same thing, except that it's solely dealing with the diffusion of water across a semi-permeable membrane from a high to low concentration. So in this example, we have this beaker of water, and we have a semi-permeable membrane separating it in half. On the left side, we have a 5% sugar solution. So those large red circles are sugar molecules. And those are too large to pass through that semi-permeable membrane, but the water molecules are able to pass through the membrane. So the left side, we have a 5% solution. The right side, we have a 15% sugar solution. So essentially what that means is on the left side, there's more water molecules per volume. And so they will want to diffuse to the other side in order to equalize that concentration difference. So if we let that beaker sit for a little while, you would actually see the water level rise on the right side as these water molecules diffused across that membrane in order to equalize the concentration of water molecules. And you can actually see this happen in a lab demonstration. So we can see here on the left side, we have water mixed with some colored molecule that can't pass through that middle membrane. And on the right side, we have pure water. And so since there's more free water on the right side, it will diffuse into that left side to try to equalize the water concentration. And then finally, we'll talk about tonicity. And tonicity is the relative solute concentration in a solution that determines which direction the water is going to want to diffuse. And so we have 3 possible scenarios here. We can have a hypotonic solution. And this means that we have a lower solute concentration. So in this picture on the left side, we're dealing with the solution, which is that blue area. And so we can see that very little of that space is composed of solutes, and most of it is water, compared to the cellular space. And so water will want to diffuse from the solution into the cell in order to equalize that water concentration. And in this example, it can actually result in the bursting of a cell if you have too much water diffusing into those cells. You can have an isotonic solution. And so this one is where you have an equal concentration of solute and water on either side of the membrane. And so you will have water moving in and out of that membrane at an equal rate. And then finally, we have a hypertonic solution, which means that we have a greater concentration of solutes as opposed to a hypotonic, which is the opposite. And so in this scenario, water will be diffusing from the cell where there's a greater water concentration into the solution in order to equalize that water concentration. And so in this scenario, this can result in the shriveling of cells as water diffuses out of them to try to equalize that concentration. And so this has implications for biological activity in that diffusion and osmosis allow for molecules and water to move in and out of the cell without the cell having to expend any energy. And this is really important for processes like gas exchange with respiration. So your cells require oxygen in order to create energy. And so that oxygen has to be able to move from your bloodstream into your cells. And then the CO2 that's produced in that process is a waste product. And so that has to be able to diffuse out of the cells into the bloodstream so that your body can get rid of it. And also, osmosis is really important for your cells maintaining the proper water balance. So water being able to move freely across those membranes is really important. And that's going to be it for this video. Hopefully that will serve as a good foundation for you guys to go on and do these lab simulations. And hopefully if you have any questions, you can reach out to us and let us know.